people ask me what I'm doing this summer, and I'm like, oh, I'm cloning DNA and I'm injecting into embryos, and they're like, what? What does that mean? So I never thought I didn't even know this was possible, to be completely honest. I didn't know it'd be, like, cloning DNA was something that a student could do. And it's surprisingly not that hard, as long as you follow the directions. But no, I didn't think I'd be doing something like this, no. My name is Teryn Coffman. My major is psychology with a minor in biology and chemistry. My research project has to do with the dopamine neurons in Daphnia magna. My funding is through URGO, and my mentor is Dr. Beckman. My research has to do with when and where the dopamine neuron cells are born in the Daphnia magna embryos. I'm trying to create like the stages of the embryos and trying to break it down into intervals so I can see, I'll be tagging the gene sequence for the dopamine, the DNA that I want, and we'll inject that into the embryos, and it'll be like a fluorescent dye. So when I stay in the embryos with like a blue dye, I should be able to see when exactly the dopamine cells were born. If we can tell when the dopamine cells are actually born, like, that's kind of a crucial piece of information, but I'm not sure where it would go from there. I know that the dopamine cells have a lot to do with Parkinson's in humans, and because Daphnia is a great model organism, it can provide some kind of insight to when they're born, what those cells do as far as like what happens after those are born. If the appendages start to grow, if they start to move, what would happen if we were to block the dopamine neuron? If we were able to like block that cell, what would happen to the motility of the animal, which could give us a lot of information, insight into humans or other animals, but I don't really know how they would go about that, because I'm not that far advanced.